Hello. Today, myself, Abdul, Muath, and Imogen will be discussing compatibility of sodium and potassium values measured by biochemistry analyzer and blood gas analyzer in different clinical situations. So, the accurate detection of abnormal levels of both sodium and potassium are essential, as these are frequently encountered in emergency departments and intensive care units. The accurate detection is very important because they are both treatable medical emergencies. Changes in these ions in the blood are categorized in two different ways, as either above or below the normal level. If blood potassium levels are above the normal range, the individual is said to have hyperkalemia, and if levels are below the normal range, they have hypokalemia. This is very similar to those with abnormal levels of sodium. If levels are above the normal range, they have hypernatremia. If they are below the normal range, they have hyponatremia. Variations in these electrolytes can be either the cause or the consequence of a variety of different disorders, and thus these levels must be accurately detected so adequate treatment can be performed. Clinicians must take care when detecting changes in potassium and sodium levels above or below the normal range, as small fluctuations can be indicators of serious health complications. For example, hyperkalemia is an early indicator of chronic kidney disease, which can lead to a variety of other serious symptoms, such as kidney failure and heart arrhythmia. Further, hypokalemia, when blood potassium levels are below the normal range, has many further complications as well. Potassium aids in electrical signaling within the heart and helps the heart to contract properly. If potassium levels fall too low, cardiac arrhythmias and dysfunctional heart muscles can manifest. Measurement of these ions are usually done in one of two ways, either by a blood gas analyzer or a biochemistry laboratory autoanalyzer. Generally, blood gas analyzers are used in emergency situations, such as in the intensive care unit of a hospital or in the emergency room, as to provide quick and appropriate results. Quick results allow for timely management and treatment of patients, and should improve outcomes due to the ease at which a clinician can make a blood measurement. Contrastingly, laboratory autoanalyzers take an extended period of time to provide results, and thus there may be a long delay between the time a test is ordered and results received by the clinician. However, due to their automation, many potential errors are eliminated, such as human error. It is common that clinicians use both methods to get a more complete picture of a patient's health. Abdul will go into more detail about these two analyzers in the methodology. There is limited data on the accuracy of blood gas analyzers in assessing electrolyte concentrations. Therefore, testing of the reliability of blood gas analyzers in different clinical situations is needed for accurate and prompt clinical decisions. There have been a number of studies regarding the accuracy of these two analyzers. A study in 2012 by Budak and colleagues conducted a similar study and found that blood gas analyzers and laboratory autoanalyzers do not yield equivalent potassium and sodium results, and that concordance between these two methods needs to be established. However, this experiment differed in that its aim was to assess compatibility of serum potassium and sodium of blood gas analyzers and laboratory autoanalyzers in different clinical situations, such as in hypokalemia, hypernatremia, and so on. Thus, the aim was to evaluate the correlation in serum sodium and potassium levels measured with blood gas analyzers and biochemistry laboratory autoanalyzer devices in the laboratory.
Now let's move to the next part of this experiment, which is material and method. They conducted a retrospective study between April 1st and 30th in an emergency department of an university hospital. They screened patients older than 18 years old whom had both venous blood gas analyzers and serum electrolytes and biochemistry panel. They attached great importance to make sure that blood samples were drawn from same vein and simultaneously to avoid improper results. The samples were collected from same vein consecutively and the blood gas injector flushed thoroughly with 1 milli solution of heparin, which was removed completely before drawing of samples. Samples collected in different time frames or from different veins were excluded from the study, and they also excluded the samples which are defined as homolyzed by a biochemistry laboratory. Then they grouped electrolyte levels of patients as hyper, hypo, and EU to compare these groups separately. All protocols was approved by the University Ethics Board. In this study, they used the radiometer APL-90 Flex machine to analyze blood gas samples, which is an automated whole blood gas analyzer with a wide range of parameters that allow to increase the portfolio of services. In just 35 seconds, the APL-90 Flex analyzer can deliver 17 parameters from a sample as small as 65 microliter. They also used the Abbott Architect C16000 to analyze venous samples. These analyzers measure substance within bodily fluids to establish a pattern of abnormalities. The wide range of tests can be grouped under several categories such as electrolytes, enzyme, blood sugar, metabolites, thyroids, hormones, lipids, therapeutic drug monitoring, and drug of abuse monitoring. This analysis can be conducted in a blood, either whole, serum, or plasma, as well as urine, semen, or cerebrospinal fluids. Both machines were calibrated on a timely and regular basis to company specifications. For the statistical analysis, descriptive statistics for the continuous variables were presented as a mean standard deviation, while count and percent for the categorical variables. Pearson correlation coefficients were calculated for determination of linear relationships between the variables. In addition to correlation analysis, linear regression analysis was performed to predict biochemical values from the blood gas values. Patients were classified into 3 subgroups by considering blood gas and biochemical values of the sodium and potassium. For the agreement between blood gas and biochemical values, kappa coefficients was computed. Statistical significance level was considered as 5%, and SPSS statistical program was used for all statistical computations.
The analysis was conducted on 109 patients, consisted of 58 male and 51 female patients, and their mean age was about 57 years old. Both potassium and sodium levels were accessible in this group. The mean level of sodium in serum samples was found to be 138.7 millimole per liter using biochemistry analyzer, while the mean of sodium in venous blood gas analyzers was about 143.2 millimole per liter, which is slightly higher. In addition, the mean variation between the two samples are minus 4.46 millimole per liter, which lie within 95% confidence interval between minus 5.25 and minus 3.67. On the other hand, the mean level of potassium in biochemistry analyzer was 4.34 millimole per liter, which compared to 3.74 millimole per liter in venous blood gas. The mean variation between the two samples was 0.6 millimole per liter, which is also lie within 95% confidence interval between 0.50 and 0.70. This will lead us to a statistical significant data. For example, a positive correlation was found to be A positive correlation was found in sodium and potassium levels in various strengths in different clinical situations. The correlation coefficient was found to be in sodium 0.852 and in potassium 0.774. Moving forward, p-values was less than 0.001 for both values, which indicates that the experiment is statistically highly significant and the null hypothesis is true. The null hypothesis is that there is a concordance between a blood gas samples results and the standard biochemistry analysis in determining sodium and potassium levels in patient serum. The percentage of predicting the variance between dependent variable, which is in our case a blood gas analyzer, from independent variables, which is biochemistry analyzer, can be determined by squaring the R. The square R for sodium serum is 72.2. In other words, this can be looked as 72.2% can explain the variance. On the other hand, the square R for potassium serum analysis is 59.9%, which doesn't explain the variance as much as in sodium. This figure summarizes the extent of the concordance between biochemistry and the blood gas analyzer in determining sodium level in the patient's serum. After measuring sodium level in 109 patients using a blood gas analyzer, patients grouped into three categories: eunatremia, hyponatremia, and hypernatremia. 68 out of 109 patients were eunatremic, 11 of them were hyponatremic, and 30 were hypernatremic. However, when the blood gas analysis results were compared between biochemistry analyzer, it was found out that 61 out of 68 were actually eunatremic, which is about 89.7% of the patients. The other 7 were all hyponatremic, which they make up of only 10% of the patients. However, for hyponatremia, biochemistry analyzer does not oppose the blood gas analysis results, as all 11 cases were found to be right. The big difference was recorded in hypernatremia cases. According to blood gas analysis results, 30 out of 109 patients were hyponatremic, but when examining these cases with biochemistry analyzer, only 10 of them were actually hypernatremic, while 19 of them were eunatremic and only one of them was hyponatremic. As a result, blood gas analysis tends to show sodium results higher than biochemistry. So if we see eunatremia or hyponatremia in a blood gas analysis, it is likely to be confirmed in biochemistry results. On the other hand, if we see hyponatremia in a blood gas analysis, it is more likely to be failed. It is more likely to fail than other group. So the percentage of agreement between the two analyzers is represented by Kappa coefficient. Kappa coefficient was recorded to be 72.43. Moving to the next slide. This is a similar figure to demonstrate the extent of the concordance between biochemistry and the blood gas analyzer in determining potassium level in patient serum. The results of the blood gas analysis level of potassium in serum was 60 patients were eukakalemic, 35 were hypokalemic, and 14 were hyperkalemic. However, when the blood gas analysis results were compared with biochemistry analyzer, 44 of 60, which is about 73.3, were eukakalemic. However, 25 of them were actually hyperkalemic, which make them about 15 patients, and only one of them was hyperkalemic. For hypokalemia cases, the numbers seem to be inaccurate because when the same patients have been re-examined by biochemistry analyzer, the number of cases was found to be only 6, which is about 17% of the patients. However, 27 out of 35 were actually eukakalemic, and 3 of them were hyperkalemic. On the other hand, hyperkalemic group, about 85% of the patient were found to be actually hyperkalemic, which is high, which they make up of 12 patients. The other two were diagnosed to be eukalemic. As it can be seen from the figure, blood gas analysis tends to measure potassium level lower than biochemistry analyzer. Also, if we can see eukalemia or hyperkalemia in the blood gas analysis, it is more likely to be confirmed in biochemistry results than hypokalemia. A Kappa coefficient of sodium is higher than potassium group, 72.43 vs 38.5 consecutively, so blood gas analysis has a higher likelihood for sodium level.
The aim of this study was to evaluate the correlation of serum sodium and potassium levels measured with a blood gas analyzer and a biochemistry analyzer in the laboratory. It was found that mean sodium levels detected in patients tended towards skewing higher when using a blood gas analyzer in comparison to a biochemical autoanalyzer, while mean levels of potassium were found to skew lower when detected using a blood gas analyzer in comparison to a biochemical autoanalyzer. As serum electrolyte levels are important in determining patient health and appropriate treatment, accurate analysis of sodium and potassium levels are crucial. The U.S. Clinical Laboratory Improvement Amendment accepts the difference of plus or minus 4 millimoles per litre and plus or minus 0.5 millimoles per litre for levels of sodium and potassium respectively to be considered the gold standard of calibration results. Seeing as the variance in mean sodium levels was 4.46 millimoles per litre, this falls slightly outside of the acceptable range. For mean potassium levels, the difference is also slightly higher at 0.6 millimoles per litre. However, the correlation values from comparing the results of the two different modes of analysis for both sodium and potassium levels were quite high, at R equals 0.852 and R equals 0.774 respectively. This indicates a positive correlation and an imperfect but not overwhelming variance. Nevertheless, the discrepancies and their consequences in clinical settings must be considered. While only marginally out of the acceptable ranges for gold standard diagnosis, this small difference can be detrimental in getting proper treatment for a patient, especially when considering potassium levels. The conditions related to lowered or elevated sodium levels require less urgent care than those related to lowered or elevated potassium levels in patient sera. When severe, hyperkalemia requires urgent medical care as it can cause lethal heart arrhythmia. On the other end of the spectrum, severe hyperkalemia can be indicative of chronic renal failure, hyperaldosteronism, and massive tissue breakdown. Both of these conditions require early and accurate diagnosis to manage the symptoms and prevent escalation into lethal territory. Thus, the 0.6 millimole per litre discrepancy between blood gas analysis and biochemistry laboratory analysis is detrimental, as some patients would not be able to wait for laboratory results. Complications related to hyponatremia and hypernatremia can also be fatal. However, these conditions tend to be more common and easily treatable, so some time can be taken to determine the results. Other studies comparing the results of sodium and potassium levels using blood gas analysis and biochemistry laboratory analysis found variations in the levels of error compared to this experiment. Bloom et al. Found a 3.36 millimole per litre difference for sodium and a 0.46 millimole per litre difference for potassium between two different modes of analysis. In both cases, these results are within the U.S. Clinical Laboratory Improvement Amendment's limits on gold standard calibration, thus being accurate enough to base a diagnosis on. Budak et al. Found the difference between sodium levels to be 4.9 millimoles per litre and potassium levels as 0.25 millimoles per litre. In this case, the margin of error is a bit higher than what was found in this experiment for mean sodium levels, and therefore considered even more out of the determined acceptable range. Potassium levels, however, were lower than the stated limits than those detailed in this experiment, suggesting them to be even more accurate in diagnosing conditions related to lowered or elevated levels of potassium. Kozaki et al. Found a difference between sodium levels of 7.9 millimoles per litre and potassium levels of 0.5 millimoles per litre, meaning the potassium results again were within the acceptable limits, while the sodium levels were quite a bit higher than the limit. In all experiments, by and large, the sodium levels were found to have more discrepancies than that of potassium, which generally were within or not so far from the acceptable limit. This is significant as it was previously stated that conditions such as hyperkalemia and hypokalemia require faster testing. Therefore, the minimal amount of error between the results of a blood gas analyzer and a biochemistry autoanalyzer is beneficial. The different values found in the results of this experiment and the others may relate to the exact machines used to make the analyses. While the blood gas analyzer used in this experiment was a Radiometer AB190 Flex machine, and the biochemistry autoanalyzer was an Abbott Architect C16000, the exact models and manufacturers of the machines used in the other three studies are not specified. Therefore, the variation in values could be due to a difference in manufacture. Also, in this experiment it was specified that all samples were drawn from the same vein simultaneously to avoid improper results, while this is also left unspecified in the other studies. If more specificity was provided in the other studies, more comparison between the methods could be made and discussion furthered. A fourth study conducted by Yeltsin and Solak only measured the sodium levels as determining hyponatremia, eunatremia, or hypernatremia, finding biochemistry laboratory analysis to measure serum sodium higher than what is detected using a blood gas analyzer in most patients, which is opposite to what was found in this study. This suggests that the previous conditions in regard to difference in manufacture may be at play in explaining the discrepancy between experiments. Solak's study would also be more relevant in comparison if potassium levels were also tested, as there would be comparison of the two studies across the board to see if mean potassium levels were also different. As previously mentioned, the methods employed in this experiment detailed that all samples should be taken from patients using the same vein simultaneously, and all samples that did not follow these requirements were disregarded. While this does help to ensure quality control and validity of the experiment, a large number of cases were left out of the analysis, dropping the number of participants from 156 to 109. Having more cases would make results more reliable, so perhaps starting out with more participants would have been beneficial, as it is assumed that a smaller portion would have been disregarded. It is also stated in the methods that only 2 exact machines were used to run all samples, as this would limit the amount of error not explained by discrepancies between the 2 machines themselves. However, this also lowers the reliability of the study itself, as one of the machines could have been faulty or calibrated incorrectly. Having the same serum analyzed by different machines would serve to further determine the extent to which the variation between blood gas analysis and biochemistry laboratory analysis results occurs. While there could be many sources of error with more machines in play, it would further the discussion of potential reasons for discrepancy and its relevance to real-life clinical settings. To conclude, this experiment used the serum of 109 different patients all over the age of 18 to determine sodium and potassium levels using both blood gas analysis and a biochemistry laboratory analyzer. The differences found between the 2 machines were marginally outside of the acceptable limit determined by the US Clinical Laboratory Improvement Amendment, with many other studies conducted corroborating this fact. By and large, sodium level variance tended towards being more outside the limit than those of potassium levels. The causes of these discrepancies could be due to a number of reasons, including differences in machine manufacturer or experimental methods. This experiment could be improved by having more participants to begin with and using more than one machine in each category. Overall, blood gas analysis is quicker in returning the results for sodium and potassium levels in sera, but less accurate than biochemistry laboratory analysis, which is more time-consuming. As a result of the positive correlation between the 2 modes of analysis, however, it is found that blood gas analysis can be used to help guide physicians in emergent conditions despite the discrepancies. Thank you.